Okay, so second part. So now we move into memory representations. So this is what I showed you before, the model before. So and then in the first part, we focus more on these interactions between different regions. So now let's look at what happened inside these regions. So these are suppose the place where we have representations, right? The memory representations. So first I will talk about memory traces and reactivation theory. Then I was going to give an introduction about MBPA and RSA, but I think you heard enough yesterday about these methods, right, that I can skip that for sake of time. So, and then I will present again 3 studies from my lab. One focusing on encoding retrieval similarity for scene reactivation. Now then looking at encoding and retrieval of perceptual and conceptual representations. And finally one looking at encoding repetitions and gist versus event-specific representations. Okay, so at the cellular level, we assume that memory representations are a group of neurons that fire together, right? So you probably know that, the standard idea is that through Hebbian learning, the neurons that fire together wire together, right? So then these experiences that happen, for example, if you're here and these simultaneously the representations for different aspects of the room and etc. situation are being active together, then they tend to be bound as a memory trace. And there has to be some, for a long term effects, there has to be some synaptic protein synthesis and some morphological changes in the synapses. So these, for example, changes in the dendritic spines and some changes here like in the synapses. So I want to talk to one of the presenters who have a different opinion, right, on the memory trace. So I actually wanted to go to your poster, but was full of people. So, but let's talk later. I want to know what are the new views about this. So, so this is kind of how the reactivation theory is assumed to work. Let's say that you have this pen for a while, and then you develop a representation of this pen that will be here a group of neurons. These are with each one with a few connections, just 4, so that are being active during time, right? So after kind of seeing that pen or using that pen, the idea is that there are synaptic changes between the neurons that during consolidation will form, creating a memory trace, right? So the neurons are not acting anymore, but the connections between have changed. So later, after a delay, when you see, let's say, another pen, and some feature of it reminds you of the other pen, that may activate as part of the memory trace, which leads to the reactivation of the whole trace, leading to the experience of remembering the previous pen, right? So this is the standard theory how this works. So there is evidence from different methods. From single neurons. For example, this famous study by Sakai and Miyashita that they looked at pair recall of these symbols, and then they found neurons that, for example, were responding to one to the, so this is a cue and this is the response. So during the paradigm, they were presented, after they learned this pair, overtrained on each of these pairs, then they were presented the first element of the pair, and then after delay, they had to pick between the 2 options, the correct one and an incorrect one. So let's say what happened with the neuron that actually represents the correct response. They found that actually starts firing before you actually make the choice, which suggests that this information is being recalled, right? So this neuron becomes active as the monkey is recalling the corresponding element. So this will be reactivation at the neuronal level. In imaging, we use a different type of method. We typically present information such as words, for example, banjo, train, paired with different contexts. Let's say, for example, a visual context, like it could be a picture, or an auditory context, such as the sound of a train. And then during retrieval, we typically present the stimulus without the context. So, and then we can see whether some of this context is reactivated during retrieval. So if you see in this study, for example, this is a classic kind of a Wheeler and Wagner study. They found that words that were studied with pictures elicited greater activity in visual regions, whereas words that were studied with sound elicited greater activity in auditory regions. Since the stimuli during retrieval are identical. The only explanation of why you would get these differences is because the participants are retrieving the associated context. So these regions are assumed to be supporting the recovery of those encoding contexts, so the reactivation. So most studies have used univariate measures. So, and there are many nice reviews like Dunker and Abelson that you can read. They have looked at a variety of stimulus association. For example, how people, reactivation of left versus right visual fields, reactivation of ventral versus dorsal streams, reactivations of auditory versus visual associations, such as these are also in Lars Nyberg, kind of early study, reactivations of faces, words, and scenes, reactivations of emotional associations, and a variety of conditions. So there also have been studies looking at the reactivation of strategic processing. So not only you may remember some visual or some sensory context, but also you may reactivate the particular task that you did in the stimulus. Let's say that in one task, for example, in that study by Johnson and Ragg, people were presented with words and they either had to make a sentence with a word or imagine the word and the associated object in a scene, right? So then at test they were presented the words and they had to remember and they showed reactivation in language-related regions for the sentence or visual-related regions for the scene. There are also studies on enactment in which, for example, that study by Lars Nyberg, they had people actually make motor movements for the, or actually that was done in the scanner, so they were actually to imagine. So you got like a, for example, a sentence, put the cup on the table, and either they had to imagine the motor action of putting the cup on the table or just doing some type of semantic processing. And they found that at retrieval, if they were actually doing these mental motor imagery, they actually were showing more activity in motor cortex. So this is kind of, it's not in the stimulus per se that you're presented, but more in the mental processes that happen during encoding. So there are many open questions about the reactivation. For example, there is still not enough evidence of how the reactivation relates to the amount of information retrieved. So the assumption is that, okay, so if there is a greater reactivation, that means you're retrieving more information, then your performance should be better, but the data is still kind of weak. Then what happened during false memories? So are the reactivations tracking true memories only, or they also happens for false memory? And a study by Kant, for example, found that they asked people to encode words, either to make a phonological judgment, I think was to sound the word backwards, or to make imagine the scene for associated with the word. And then at test, they had to recognize the word and rate the recollection. And they found that even when people, rate what of the context, when people made a false alarm, it's a new word and they thought it was old, and actually attributed to a context which was impossible because the word was old, still they show the reactivation for that context. So if they saw, oh yeah, this word I saw them and I did an imagery, in the imagery task, they would show kind of visual cortex activity as if they had been in that condition. So the idea is that then perhaps some of these activations may be produced top-down, right? So this is one of the most tricky issues in reactivation. Are these sensory reactivations during retrieval reflect a bottom-up spontaneous process, right? Or that these regions are kind of activated by the memory trace directly, and that's causing you to think about the event? Or is that you are trying to retrieve and then start focusing on the information that you expect, and then you activate those regions top-down? So this is then the relation between reactivation and subjective recollection is also other issues. So in a standard, just a brief about univariate versus multivariate analysis. So as you know, in univariate, we compare the means of a group of voxels. Let's say this, in condition B, there is a greater mean for these 9 voxels that in that condition. And so multivariate activation analysis measure the spatial distribution of activity across voxels, and importantly, they can detect differences when the mean activity is the same. Let's say condition C has the same mean activity, you see it's 2, but you can see a different pattern. So because it's based on the correlation is negative actually, minus 0.3. but also can detect similarities when activity is different. For example, C is more than B, you can say it's different, but actually the correlation is 0.9, right? As you can see, there is a very similar pattern, even though there is a difference in activity. So clearly the correlations give you different information than the means, right? So you can detect changes in multivariate patterns that you cannot detect with univariate activity. So the main two methods are multivariate pattern analysis, in which you train a classifier to detect these different patterns and be able to classify, let's say this is for condition B and condition C, and kind of and representational similarity analysis, in which you basically look at the correlation between a different type of stimulus or different types of conditions, and you heard a lot yesterday from. So now the introduction is shorter because I don't want to go over time again, and you heard a lot about RSA yesterday, right? You don't need, I think, more. So we use a representational similarity analysis, but we use them in a different way. So rather than looking at necessarily differences across stimuli, for example, Christian was showing yesterday, okay, so let's say that you have the items that were kind of associated together versus the ones that were not, or faces and house, or the scenes, for example. We typically look at the similarity between encoding and retrieval. So another thing that we do is also not only look at similarity at the set level, but also at the item level. So in previous studies, the reactivation has been studied at the set level. So as what I mean by set is for a group of items. Let's say that you find that there is more visual cortex activity for words that were paired with pictures than for words that were paired with sound. This is not a one particular word or one particular picture, it's a whole set of stimuli, right? So this is the way they are typically done. You have a group of items encoded in context A, another group of items encoded in context B, and when you present the items, you see whether the reactivation of context A versus B. However, in real life, right, in episodic memory operates at the item level, at the particular event specific, right? It's not enough for you to remember that you came by car today as opposed to take the bus. You need to remember where you parked your car, right? A specific location. It's not enough for you to know that you had ice cream as opposed to having beer, but you may want to remember what specifically flavor of ice cream you had, right? So these days, if we want to get to the item specific level, we need to use a different method that is not collapsing across all items, but actually are focusing on individual items. So what we do to do that is actually we compare the similarity between each item during encoding and each item during retrieval, and look at those at the item level. So let me show you this first study. In the first study, we had participants. I think that, well, that was a paper that I sent. I don't know how many of you raised your hands if you had a chance of looking at that. That's a WING study. So this, in that study, the participants during encoding saw pictures paired with a label, like let's say ocean wave. And we use these pictures that develop in MIT, but Odoliva's lab, that have hundreds of scenes that are very stereotypical, right? So people immediately know what the labels are, like there are golf courses, for example, barns or tunnels or kind of forests or bathrooms or things like that immediately people will say, oh yeah, that's a bathroom, or this is a barn, right? So they are stereotypical, and then you can get many similar ones, which is important. So then we presented them with a label. People rate the representativeness of the image for the label. And then during retrieval, we presented the label alone, right? This is critical for study reactivation. So without the picture, people remember the picture, right? That was during encoding, and rated the vividness of the image from 1 to 4. So we wanted to confirm that actually those vividness ratings actually were a good memory rating. So as I mentioned before, to do that, after scanning, we had a forced alternative, forced choice paradigm, in which for each picture we presented the original picture with 3 distractors, and participants had to pick the original picture and rate their confidence from 1 to 4. So then, as you see, the pictures are very similar. So actually remembering requires very kind of a great detail to know which particulars you saw. So then we wanted to confirm that those vividness ratings in the scanner were actually predictive of that accuracy, and this is what we found. So if you look at the 4 levels of vividness, and here I call M for memory 1, memory 2, memory 3, memory 4, and you see that the misses in the test goes down, whereas the high confidence hits, right, basically go linearly up. So basically these vividness ratings are strongly correlated with memory, so we use those ratings instead of the post-scan measures to measure reactivation, because the problem with using this test is that it's partly determined by the distractors, right? So we wanted to have a measure that is primarily driven by the single item, right? So the method that basically what we look is the encoding retrieval similarity and look at the effect of specificity. So imagine that you have, during encoding, you see a picture of an office, and you imagine that then the hippocampus is activated, and then there is a representation in the cortex that is linked to the hippocampus where you store that image. During retrieval, you get the word office, and then you reactivate in interaction with the hippocampus, you reactivate those voxels. Our assumption is that if your memory is better first assumption is that these patterns will be specific to the picture, so they will be different for the office than for, let's say, this skin scene, and that the encoding retrieval similarity will be associated with memory. So high encoding retrieval similarity will be associated with good memory, and low encoding retrieval similarity with poor memory. So for those that have a cognitive memory background, you may remember kind of encoding specificity idea, or the transfer appropriate processing is a strong idea in memory research, in which assumes that memory is a function of the similarity between encoding and retrieval operations, or between kind of the reactivation of the encoding context in the case of encoding specificity. So this is partly in the way that we think that encoding specificity or transfer appropriate will be represented in the brain. Basically, if your brain becomes to be in the same state that it was during encoding, then your memory will be good. So critically, as I mentioned, we distinguish between item level and set level, and to do that, we look at what we call item level is basically the similarity between the encoding pattern for each picture with the encoding pattern during retrieval, right, 1-to-1. And the set level is basically the comparisons across all the other pictures, right? So this will give you, the set level is basically gives you an overall level of similarity, which provides a kind of like the baseline for us. So our assumption is that to the extent that a region, that in a particular brain region, item level similarity is greater than set level similarity, then this region is storing picture-specific memory traces, right? Otherwise, it wouldn't be different when you look at only 1-to-1 than across trials. So is that clear? Because that is essential for the rest of so the item versus set level is the way that when we look at the specificity, the extent to which the 1-to-1 is better than the cross, than the off diagonal, right? So basically we look at the diagonal for encoding retrieval versus the off diagonal. Actually going to the diagonals now. So, I mean, we're talking about, actually, it was a question about the Searchlight yesterday. So we use a Searchlight procedure. So basically, let's say for encoding, we have barn during encoding and barn during retrieval. We take a particular voxel, and around the voxel, we take a 5x5 cube, which we make vectorize, calculate the correlation between those, and put it in that voxel, and then move the Searchlight around the brain, and that covering basically the whole brain. So each voxel, instead of containing an activation, contains the correlation between encoding and retrieval for the voxels surrounding it, right? So for the 5x5 voxel cube around it. So then this is for the item set, right? So this in green here will be for all for 1-to-1, and those will be the diagonal. And then we separate we look at for the fourth level of memory, right? Memory 1, 2, 3, 4. And we only compare the set level within the same level of memory to avoid confounds in terms of the overall memory, right? So we want to say, for the same level of memory, what's the difference between 1-to-1 versus across, right? Because it could be just general levels of attention or something like that across one level of memory that is not the critical point across for the. So then we do the same for set ERS. In this case, we compare across different pictures, look for that, and then end up filling the whole diagonal, right, for this set, right? So let's say that this is tunnel, and this will be the whole diagonal with tunnel, with barn, tunnel with ocean wave, tunnel with it, right? So across the whole diagonal, and that gives us kind of the rest of the square. Is that clear, the method? So basically, we compare the diagonal of the correlation matrix to the whole matrix. So this is what we do, we compare those. So then we do a two-way ANOVA in which we compare the main effect of a specificity is to what regions where there is greater encoding retrieval similarity for item level similarity than for set level similarity. So these will be regions in which the activation pattern is more similar for the same pictures and for different pictures, but they don't have an impact on memory, right? So they are not directly influencing your memory judgments. So then we have a look at the main effects of memory. These are regions in which the encoding retrieval similarity increases with memory, greater similarity but greater memory, but they don't differ when it's item to item than across. That will be kind of like a general similarity across pictures that is not sensitive to individual pictures. Is that clear, the idea of a memory main effect? And finally, our goal is to study the memory by specificity interactions, which are the regions in which the similarity contributes to memory, but only when you look at the item level. And those are the ones we think are the ones storing episodic memory representations, right? Because those are the ones in which your memory is better, but only when your pattern is for one specific picture, only when that representation matches the encoding similarity. Is that clear, the idea? Okay. So what we found is first in terms of main effect of specificity, we found that the retrospinal cortex was showing this kind of greater similarity for item than for set, but didn't differ with memory. So how is it possible that a region, right, is very sensitive to actually the pattern of voxels, they're very sensitive to the same pictures, but they are not actually influencing memory? And our interpretation for that, and this was kind of post hoc account, is that possibly this region is actually mediating semantic memory. Since we use stereotypical scenes, such as barn and bowling alley and ocean wave or things like that, you can still imagine the scene even if you don't remember it, right? So if I say, okay, imagine a tunnel, you can make an image of a tunnel even if you don't remember the specific picture during encoding. The better you make an image of the tunnel, the more likely it's going to be similar to the picture of the tunnel during encoding, but that doesn't help you to remember whether you saw that particular picture during encoding, right? Because you basically can do that for any picture. You see what I'm saying? So that will give you better high because tunnel to tunnel is more similar than to tunnel to barn, right? So you are making a similar picture, but that doesn't tell you that you actually saw a tunnel, right? So this was our interpretation, and so and this fits with the idea that this region is part of what Moshe Bar called a context frame, which is a group of regions that actually store abstract representation of context, which actually are things like that. I mean, typical. It will be, as you know, I mean, we have a concept of a car, a concept of a dog, and a concept of a table, but we also have a concept of a classroom, a concept of, of scenes. And according to Moshe Bar, this network that includes the parahippocampal cortex, retrospinal cortex, are mediating that context frame representation. So that fits very well with that. But we wanted to test this idea. So we went back, and this is going to be a separate study. We looked at and this is more similar to what is done with RSA in domains like semantic memory, in which you have different representational dissimilarity matrices that represents a model about the stimuli, right? Basically, how would how the similarities will be based according to visual properties or according to semantic properties. So what we did is, first we look at, for visual, we use what's known as the Gabor-Jet model, which basically measures all the frequencies across the images and tell you, for example, that these two images are different. For example, have a low Gabor-Jet similarity because have mainly vertical lines or linear, but this has more circulars or so forth. Whereas, for example, this will be similar, I don't know, to a wheel of a car. So basically, this automatically tells you similarity in the low-level frequency analysis. And when we apply that Gabor-Jet representational dissimilarity matrices to the encoding data, so that's independent of the retrieval, because that will be just across the images, we get primary visual cortex, right? Consistent with the idea that this is a region that is where similarity, neural similarity, matches with similarity in perception. Then we wanted to look at semantic content. So what we did is analyze each of the images in terms of 500 elements like sky, windows, trees, mountains, chairs, etc. And then we can calculate a matrix about the similarity with all pairs in the 96 images we had, and this is it. And something that confirms that actually this makes sense is that you can see eyeball that the indoor pictures are more similar to indoor pictures and the outdoor pictures to outdoor pictures. Obviously, the outdoor pictures have commonalities in trees and sky and things like that, and indoors. So then when we apply the similarity matrix to the encoding data, to the similarity across the pictures during encoding, what we find is retrosplenial cortex. So the same region that we found to have this specificity. So basically this region is kind of tracking the similarity across the semantic component. So a test when you remember, oh yeah, did I see a bowling alley? I say, oh yeah, I remember, or let's say, did I see a living room? Oh yeah, I remember seeing did I see a living room? And you imagine, okay, was there a table and a sofa? And I think you start kind of basically finding similarities with other pictures that have sofas and tables and so forth, right? So that explains why they're more similar to to the same picture than to the different pictures. Okay, so the second main effect was memory, right? And the memory here increases for both the frontal regions, show the similarity that increase with memory level, but didn't differ for item than set. So this is also intriguing, right? Because in this region similarity there is greater encoding retrieval similarity as a function of memory for the ones you remember better than less, but it doesn't differ whether you're looking one-to-one versus across pictures. So it has to be some function of this region, right? That is actually when you use this region in the same way during the retrieval, your memory performance is better, but that process is not specific to one picture. So it could be just a general picture processing or a general, right, a control process that helps you process mental image or visual processing, that if you apply it again in the same way during the retrieval, your memory is going to be good, but it's not specific to each picture, right? So, what I found interesting about that is that we have the tradition of thinking that the representations are in the back in the brain and the frontoparietal regions mediate processes, right? So we have this distinction between processes and representations that we inherited from cognitive psychology, right? In cognitive psychology, it's easier to define representations and processes because representations are the things inside the boxes and the processes are the arrows, right, that happens between the boxes. So the idea you have a representation, you do something with it and you pass it to another box and do something else. So I think that basically our model is like a cooking model, right? So representations are like the ingredients, right? And then the processes are all these kind of, you can peel and slice and grate and pour and mix and whisk and boil and fry and roll and stir. Sorry, but all those, you apply them to something and you can apply it to something else, right? So the idea is that processes are independent of the representation. You can have attention, top-down attention to a house or top-down attention but when you get to the brain, things are not like that, right? So first, the distinction is not clear in the brain because processes are representations, right? So what is a process? It's a group of neurons firing together. So what is a representation? It's also a group of neurons firing together. So how do you distinguish that? So representations are processes also because the representations do not exist really until they are actually firing. So has to be active, so they have to be a process of being reactivated. So they are not really necessarily fixed things that exist outside of the process, right? So what I was trying to wrap my mind, so these are the type of things that will need a completely new vocabulary, right? If we want to start linking cognitive psychology concepts to neuroscience because the standard concepts that we had don't fit the brain. So, if you want to think about the cooking metaphor, right? You have to think that you don't have a knife, really, a cutting that works in general is a way of cutting that is only specific to carrots, right? So and then you have an orange knife that applies to carrots, and then everything blends together, and then you kind of, you know that if you are cutting a carrot with a knife or the carrot is being, basically you start kind of breaking down the distinction. So we'd say, okay, well, the idea is that these frontal regions have a representation, right? In a way, that way of operating the process on the posterior region that helps memory has a structure, a certain distribution of voxels that is very typical. It's not that the whole region is active, because we are not looking at mean activity, right? So we are talking at one possible argument here is that because we are using an ROI, right? Or no ROI, but we are moving the Searchlight, that we really are combining different regions here, and they're really kind of different sub-regions here, that when you split them, then you'll see. Anyway, so I thought that this is an interesting for the future, and to try to understand how representations relate to processes and vice versa. But finally, the main finding of the study was this interaction, right, in which we get regions where encoding retrieval similarity increases as a function of memory, but only for item and not for set ERS. That means that this region is only helping memory to the extent that when your pattern of activity retrieval matches the encoding, and for that particular picture, right? So this is, for me, is the key for describing a memory representation. So I would say that it makes sense to argue that this is one of the regions where the picture memory traces were stored. Now, you would argue, well, we're talking about the distribution of representations of the cortex, right? So how is it possible that you find just a little region there, right? So this seems to be incompatible with the idea of a distributed representation. So I think the reason why this is the tip of the iceberg, right? So this is one of the regions that came out significant for that interaction. But the other problem is that the same way that we do this is, we're looking for a region where the similarity between encoding and retrieval is associated with memory for all pictures. So in a way, we are kind of, it's a bit ironic that we expect a region that is sensitive to individual pictures will be sensitive to individual differences for all the sets of pictures, right? So that I think is one of the reasons. So there could be another set of regions that are specific, that is very sensitive to individual pictures, but only for the outdoor pictures, right? And another group of regions that will be sensitive to things for the individual, but only for the indoor pictures. So there could be the regions that are sensitive to trees, really will be sensitive to individual features of that image only for the tree pictures, right? So in a way, this is kind of only looking for regions that are at the same time very specific to individual pictures, but also sensitive for all types of pictures. So this is, we are trying to get beyond that by starting to looking at encoding retrieval similarity, item specific, but for different sets of pictures. So in the next studies, we're moving to objects in which we can have different categories, right? Fruits and kind of trees and so forth, so that we can start looking whether you get both item specificity, but also differences across categories. Anyway, so just to show you what's happening here, so basically you have a group of voxels that show difference across pictures, and when those voxels are similarly reactivated during retrieval, you get a high rating, and this is an example. These are actually voxels from that region, and these are actual pictures, but just to illustrate what's happening, that when you actually look at high correlation, you can eyeball that this was kind of one of the 4 ratings, that is greater similarity. So you can see that. Okay, so then the model I mentioned is not only the cortex, it has to be connected to the hippocampus, right? So we assume that unlike other types of memory, like implicit memory that do not require the hippocampus, episodic memory requires the hippocampus to integrate these cortical traces, right? So to actually allow us to have consciousness of the past event. So how can we look at that? So what we did is then look at the regions where we found that the item-specific encoding retrieval similarity for successful memories and link them to functional connectivity with the hippocampus. So in one earlier study, so we found that, for example, that particular region, when we look at regions that where activity varies as a function of similarity, we found the hippocampus. So this case was during encoding. So greater activity during encoding in the hippocampus was associated with greater encoding retrieval similarity in that occipitotemporal region. But we also found it during retrieval in a previous study, which I didn't mention before, And in this case, we use a mediational analysis, and we found that the effects of encoding retrieval similarity on memory was mediated by hippocampal activity, which is consistent with the idea that the hippocampus is necessary for that cortical reactivation to affect memory. The hippocampus has to be activated too. So, I mean, it will be interesting, and we are thinking on doing that, looking at implicit memory, because I expect that for implicit memory, cortical similarity may be able to produce priming, even if the hippocampus is not activated, because it kind of that's a match with a cortical representation independently of the medial temporal lobe. Okay, turn to the second study. As I mentioned before, I mean, the pictures are great because they produce great, I mean, the encoding retrieval similarity, because they are so complex. Great, I mean, you have many reasons to find kind of differences across these different pictures, but the problem is that because they are complex, you don't know what's producing the similarity, right? And also, you eventually want to analyze the different components of similarity. Are the pictures similar in a perceptual way or in a conceptual way? We did it by analyzing Gabor jet or by doing just the overlapping objects, but I mean, it's complex. So we decided to shift now to object pictures. And we are working on it on this object database. We have over 1,000 objects that have been kind of divided by, we have living, non-living large, and non-living small, because there is evidence that actually the size matter for large and small non-living objects for all these different categories. And we have a variety of measures, normative measures about like name agreement, COCA frequency, manipulability, and so forth. Visual properties like Hmax, that is a measure of how that picture will be represented in visual cortex, image energy, RGB values, and also semantic properties. And this is the advantage of the objects, because there have been a lot of norms for similarities across these different objects in terms of their concept, right? So like a duck and a dog are both living, right? But they are different. A dog is more similar to a cat because it has 4 legs, and right? So it's a mammal. So there are all these differences that can have been normed, and there are some norms done with words, just presenting words and saying, basically, list all the semantic features, right? And then looking at the correlation matrix on those features. We are doing the same, but with pictures. So we have been doing a massive kind of MTurk normative study in which we present the pictures and people read their norms. So this is an example of the visual model for those using Gabor jet again, which is a good measure for us. And the semantic model is just the overlap in semantic features. And it's nice that within that semantic model you can already see the categories, right? So these are sorted by the categories. You can see these little squares corresponds to things like fish or furniture or things like that, right? So we did a study based on an old study by Barrack and Butcher from 68, and I always think it's good to go back because, I mean, there's so many rich studies done in memory psychology in the 60s and 70s. And in this study, for example, they found that when you present objects and then test memory for the objects, either with a purely verbal test in which you ask people to recall the names of the objects, or in a purely visual test in which you present 2 pictures of the objects, one, and they have to pick the one, which is really dependent on visual memory because you know that they're all, it's only visual memory. Actually, the performance between these 2 tests is not correlated, which is really striking, right? Because you expect that if you have a common representation, then the memory test should be correlated. But here, they actually were not, which supports the idea that there are separate representations for the concept of the event and for the visual part of the event. So we tried to look at that. So we presented all these objects with the initial, and the task where participants was to press a key when the letter didn't match the object. So this is, I guess it's cardinal and glasses and bench, they work, but sometimes it will be once in a while in which it doesn't work. So we wanted to ensure that people will be naming the object, which is important for a full semantic processing of that object. And then after retrieval, after the encoding, we had in the scanner one test, and then outside the scanner another test. First, they had for all the objects a conceptual test in which they got the word, and they had to say from definitely, from definitely new to definitely old. And then after scanning, they got a perceptual task in which we didn't use forced choice, but because we wanted to get individual measures, but we had very similar very similar pictures. For example, here's another glass in which they had to answer new, right? That means that the answer of whether it's an old or new picture has to be really detailed. So this is measuring, and this is nice because you can actually do this test in sequence without affecting each other, because even if you know that you saw a bench, that doesn't tell you that that was the particular picture of a bench, and then you can link directly the visual and semantic performance at the level of the object. So when we did a first analysis just based on during kind of encoding, and you see that this is the visual model and the semantic model, and as expected, you get regions that were similarity across voxels is correlated. The second order correlation with the Gabor jet is in primary visual areas, whereas the semantic model extends more anteriorly into the ventral pathway, but also frontal areas. So this confirms that have been shown many times in semantic studies that, posterior back of the brain stores visual features, and as you move along the ventral pathway to the frontal lobes, you start storing semantic features. But our interest was whether how these semantic differences versus perceptual will predict later memory. Right? How they would relate, because both semantic research and episodic memory research are huge areas, but unfortunately haven't been as connected as they could be, right? So you probably think the same. So because we know that memory depends on we know a long time that levels of processing and semantic processing enhances episodic memory, but most work on semantics within the episodic memory has been on just rough differences in processes, right? High level, semantic versus perceptual, right? Or just levels of processing, but haven't been at the level in which people in semantic memory works, at the level of categories, at the level of features, and so forth. So first, we did what we did is look at the conjunction of different types of the visual RDM will be regions where the similarity is correlated with the visual model, the semantic regions that are correlated with the semantic model, and then we look at whether the memory was successful in these regions for the perceptual test and the conceptual test, right? Is that clear? So one analysis gives you the regions that show sensitivity to the properties of the objects, and the other factors is whether those voxels or that similarity is associated with better memory in the two tasks. So first, let's look at the quadruple conjunction, right? In what regions really they are sensitive to both visual and semantic and predicts memory in both tasks. And not surprisingly, we get a medial temporal lobe region, here most in the parahippocampal gyrus, and you can see here that the similarity really is greater for subsequently remember than forgotten, both for regions associated with the semantic model and visual model, and both for the conceptual and perceptual tests, right? So these are regions that basically store everything about the objects and predict memory in general. So, but we found also the regions that when we look only at the conceptual areas, we found that one particular region predicted memory both during encoding and during retrieval. So these pattern or these regions were also showing differences during retrieval. Consistent with the reactivation patterns of the same voxels during retrieval for hits and misses. So this goes beyond the previous studies in which we show the reactivation of the region during retrieval, like that occipital, because it's actually showing that that's purely conceptual, right? Because these are voxels that were correlated with the conceptual RDM. So it's really a conceptual reactivation. Then we did the same analysis that we did for the other studies, right? Looking at item level versus set level ERS, and we confirmed the specificity effect that for the posterior cingulate, right? So greater specificity for item level than for set level, which in a way suggests that perhaps it's not specific to a scenes as we previously thought, right? I mentioned the Mosher-Bar idea that retrosplenial, because these are objects, so there seems to be a more general semantic role for whether you are remembering the concept. So, and in contrast, dorsal PFC, where it was generally in the memory effect, but it's the same for item and level. So again, this is the question of whether this is a process or a representation, right? So it seems that both the frontal in the previous studies and the parietal regions are showing that memory-related effect, but do not distinguish between one-to-one or cross different representations. So, but still there is at the level of similarity patterns, so suggesting perhaps a rolling representation. Finally, when we look at the critical interactions, we get not only posterior regions, right, but also anterior regions, like the frontal lobe. So this is interesting, right? Because the left frontal cortex is a region that we know is sensitive to differences across objects. And in this case, we are showing that actually similarity. So in a way, that pattern that you activated for, let's say, when you see an apple, and you are as a pattern within the left inferior frontal cortex, if you kind of reactivate that semantic specific pattern during retrieval, then you get greater similarity, and it's specific for each object. So, okay. So turning to the final one, so here is so I have 10 minutes. So in the final one, we look at the repetition, right? So in most situations, I mean, in real life, we don't see just one object of each kind, right? So we see many different chairs and many different, and there are many memory processes that happen through repetition. I mean, one is the concept formation, right? That's how we learn, right? That there is a concept of a dog by seeing many dogs, and that's how also through many situations we get schemas that generalize across situations, right? So this is really important, and also on the downside, that repetition can produce false memories, right? Because then we tend to create a gist representation that will kind of produce the illusion that something happened even if it did not happen. So we were interested in these different effects. So what we did is have 6 runs, and we presented these 72 types of objects, but some were presented only once. Here was, for example, like an apple, in that case, only one run. Another were presented 6 times. So basically had once versus 6 times, and the repetitions happen in different runs, just to avoid kind of short priming or things like that. So and also to control by the fact that they were similarly spaced across the 6 runs. So, and then we had again the 2 types of tests. So we had a word test in which they were kind of presented the word and had to read from definitely new to definitely old. But we also had novels. And so we had 2 types of old, the old 6x and the old 1x. They had novels, and also we had related words, right, like a scarf that was related to another item of clothing but was not seen. And then in the picture test, we also had the lures, right, in which were similar but different, right? And again, we can have lures for the 1x or for the 6x. So you can have old x6, 6x, 1x, novels, and then lures for both. So these are a similar plane to that one, but not the one you saw, a similar host to the one, right? Okay, so first analysis, let's look at the picture test. So we looked at the reactivation of the same picture. So these are, let's say that you saw 6 apples, and then you present the same apple, one of the same apples. So you can look at the retrieval of the same picture. So brain regions where there shows that one similarity. So for the same picture, then we look at the similar, in which we present a counterbalance. Let's say that picture in another subjects will look at in relations to the other pictures, right? The similarity, and then different, in which is we look at the similarity between, let's say, a teapot and apples, right? Is that clear, the logic here? So I mean, what we found is that the hippocampus was showing a pattern that resembles what has been shown as called pattern separation, right? So in the hippocampus, there was encoding retrieval similarity was greater for both the same was greater than both the similar and the different. So actually, let's say the similarity between one apple and the same apple was greater than to a similar apple, right? So these regions are actually separating between actually a similar pattern, which is similar to the logic that was used by kind of Baker and other people from Craig Stark lab, but they did it with repetition suppression, right? And just presenting the one item again. We're doing in a whole set of items and using multivariate kind of representational similarity analysis. And also we are keeping all these are old, right? So that's another thing. We're keeping constant the oldness of the items, right? So they all have been seen. It's just that we're looking at whether how we calculate the similarity. But we found other regions in which they were showing just a linear effect, what we call a similarity-related DRS. So basically is tracking similarity. So like parahippocampal cortex and the left inferior frontal gyrus. So it's interesting that the parahippocampal cortex shows similarity because it fits with the idea that the hippocampus store more item-specific, whereas the parahippocampus is more of a familiarity, just more kind of a semantic regions in that sense. So yes?
Can you tell me which is a very hippocampus? Can you separate the different?
No, unfortunately, we are using a standard resolution, right? So we use kind of a 375 isotropic. So I mean, we could try to look, but they will be without having high res. I don't think it's worth trying. So yeah, we are just starting high res studies. So, but since we are kind of looking at kind of also the whole brain here, it's more difficult to do it with a high res. But yeah, I mean, yesterday, I mean, there was amazing kind of the data for that. So I'm really inspired from yesterday going back and try to do this analysis, because even being able to look at the kind of medial and lateral interrhinal cortex, this is fantastic. So, and what we did in this study, we look at the word test, right? And then we look at when we compare just the 1x to the retrieval, and here we're using the word retrieval, so then there is no issue about which is similar, and either we compare to that to one T-pod or to the 6 T-pods in the, I mean, this will be another subject, right, because they're counterbalanced, they were the 1x or 6x, just to show you. So our prediction was that regions in which the similarity is similar to the 1x than to the 6x, what type of memory you think that will be storing? So if in a region, right, the similarity doesn't differ for the ones that is presented only once, and the one that is presented 6 times, what type of representation do you think it will be storing?
Episodic.
Episodic? But it doesn't matter which one you saw. So basically, it's not event specific. It's just kind of more of a gist level. So we assume that it will be more semantic. Whereas the one region in which show greater similarity for the 1x than for the 6x will be in which one interference is actually affecting the critical kind of one-to-one similarity for the 1x. Having seen more T-pods is actually distracting, right? Because in the other case, if it's not distracting, if there is no real interference, it's basically because you are comparing something that is common across all of them, which is only the T-pod concept, right? So this is the behavior in case you want to see. So what we found is that the left temporal cortex was showing kind of greater similarity for hits than misses, right, for both the 1x and for the 6x. So this region is basically similarities driving memory regardless of the number of times that you saw it, whereas in the hippocampus, actually we get greater similarity for the 1x than for the 6x, suggesting that region basically similarity is affected by having seen more T-pods kind of is interfering with that particular. I mean, suggests that you remember one particular event, and so having seen more events is actually disruptive in that region. So I'm sorry, I hear there's a problem with the animation. So another analysis we did is to compare each of the 6 runs. So for the 6 runs, to compare the similarity with the test. So the T-pod presented in each of the runs, so we calculated separately, and we found that in these occipital regions, basically there was like a primacy effect. So it's actually similarity, suggesting that when you think, I asked you, did you see a T-pod? You are primarily retrieving the first T-pods that you saw, right, rather than the last one. So which is interesting because we know, I mean, the primacy effect, well, I mean, of course, has been studied more in working memory, but we know primacy effects can be seen also in long-term memory. It might reflect an attention effect, but also kind of a proactive interference, right? The more that you see similar things, the less that they're stored. So these kind of are different explanations for this effect. Either proactive interference, so the first representation interference with the second one and so forth, or some attention effect. So you are attending, you see another T-pod or another T-pod, you start attending less and less, so the repeated representations are encoded more poorly, so they are not retrieved later. So, okay, so just to summarize what I've seen before, so I presented 3 studies. So one, we look at the scene reactivation, and we found that this Specificity effect was related to the specific kind of semantic components of the image. Frontal region were showing more a process-related reactivation that was independent of whether it was match one-to-one or across. The item-specific reactivation in occipitotemporal cortex correlated with activations in the hippocampus. Then we turn to conceptual versus perceptual representation. When we're looking at the conjunction, the quadruple conjunction for semantic, visual and semantic models for the conceptual and perceptual test, we found the parahippocampal cortex. Then retrospenia was replicated here, and then we also found this specific, instead of being more occipitotemporal, we got the visual and the frontal, and then these regions that are sensitive to 6X and that proactive interference. So, thank you very much, and on time.